So we then tried to see if we could actually now reintroduce these organoids, these mini guts, into the gut of a mouse, and we're now actually thinking of also doing that in patients. And what we did is in collaboration with Toshi's former lab in Tokyo, headed by Mamoru Watanabe. And what Toshi in Utrecht did is he, out of a mouse where every cell emits red light, it's very easy, you can actually track, and whenever you see a red cell, you know it comes from that particular mouse. He sorted the green stem cells, in this case from the colon, the large intestine. Here's the sorter. He went from one stem cell in culture in a few months to about 100 million cells. We can freeze them in liquid nitrogen, send them to Tokyo. There they had prepared Japanese mice that had no color, so they're black. By giving them DSS, a chemical in their drinking water, they cause ulcers like IBD, like colitis ulcerosa, which essentially are superficial damages to the epithelium. If we don't damage the epithelium, the organoids will do nothing. But if we do damage the epithelium, and what they do in Japan is, so they get our organoids that you see here, they put them in a buffer, they inject them through the anus, then close the anus with a little bit of super glue, because otherwise it will come out again for just a few hours. And during those hours, these organoids, these mini guts, will actually scan the epithelium, and whenever they see a defect, they will land there, so they see the collagen and the laminin that's under the normal epithelium. They open up and they actually seal these holes, these ulcers, and the mice actually recover much more rapidly if you give them organoids than if you don't. Now, this is an animation, as you probably realize, but this is what we see in real life. So this is one of the colons of those mice. So typically DSS makes these lesions in the distal part of the colon, which is, so this is a colon that's cut open. We look in the inside, you can see patches of essentially Dutch cells. So this is Dutch tissue, this is a Japanese colon. And this actually, we've left these mice for at least 6 months, there's still quite a few running around. They're happy, they use these cells, these tissues. And under the confocal microscope, you can see this is the transplanted tissue, this is the black subepithelium, and this is the red Dutch epithelium that was grown, expressed from a single stem cell and expanded 10 to the 8th fold. And here we see now a few other markers, so in essence, this tissue is entirely normal. People are always afraid that when you expand cells outside the body that you turn them into cancer cells. So if we then reintroduce them into mice in this case, you would cause cancer, but that turns out not to be the case, at least in these mice. Well, actually now we have a very big grant with the Children's Hospital. We can grow human colon and human small intestinal stem cells as well. And we are gearing up to make all of this, to do these other GLP conditions, good medical practice conditions, GLP, and then be able to transplant epithelium in rare genetic diseases where kids are born with a normal looking gut, but the epithelial cells have no brush borders. So they cannot digest food. They're totally dependent on intravenous alimentation. They live for 2 to 3 years and then there's no veins left and they essentially die unless they get a gut transplant. So while they're on the transplantation list, we are probably allowed to see if we can replace part of the epithelium with this epithelium that we probably obtained from one of the parents or one of the siblings. So that's an ongoing project. In the stem cell field, there is this concept that the stem cell is not out on its own, it sits in a niche. And for instance, in the bone marrow, when you get a bone marrow transplantation, the donor bone marrow is injected into the brain. It finds the long bones, it goes into the cavities in the long bones, and there are specialized sites where these stem cells will land, and then they will make blood cells for the patient. So it was always assumed that in the gut there were specialized cells that surround the bottoms of the crypts that were the niche cells of the crypt. Now in these mini guts, we started from a single epithelial cells. There are no niche cells. There's nothing that surrounds these crypts. And you might argue, well, you're giving a lot of growth factors, maybe they don't need the niche anymore. However, it doesn't explain why you have particular areas where there is a stem cell niche here and other areas where you have the differentiated cells. So the structure organizes itself into crypts and villus-like domains without any outside cues. So Toshi, again, the same Japanese postdoc, reasoned that possibly there is a niche cell produced by the stem cells. And I already mentioned the Paneth cells. So Paneth cells, you probably had histology, you might recall, are sitting at the bottom of crypts. They produce lysozyme and they produce small peptides that kill bacteria. So they are believed to be the natural immune system of the gut. So they constantly kill bacteria, try to get close to the stem cells, so that the bodyguards of the stem cells. But Toshi argued, well, maybe they're not only the bodyguards, maybe they also make the growth factors, and they're also the niche cells, so the bodies of the stem cells. And he realized that when you section with a confocal microscope through the bottoms of a large number of crypts in these mice where the stem cells are green, you can see that here, there's almost a geometrical distribution between black Paneth cells and green stem cells, as if they find each other and they need each other. Now, Toshi did a number of experiments, but the best one is this one, and I'll try to explain this. So what he did on the sorter, when we make our cell suspensions from crypts and we sort out the stem cells, he realized that we have to be fast because epithelial cells do not like to be outside the epithelial context. They very often immediately die. So he makes a crude suspension, immediately runs it through the machine, and then starts culturing the cells. And the consequence is that there are very few droplets that have a single cell, and they are in this particular case here. If you gauge here on forward and side scatter for the insiders, the droplets will have at least 2 cells. And if you gauge here, you have many, many cells in one droplet. So we usually throw them out. But in this case, Toshi compared the single cell droplets with the double cell droplets. And on the single cell, in green, these are the stem cells. In red, these are the Paneth cells, stated for CD24. And there are no droplets where you have both green and red in the same droplet, because there's only one cell. And you're either a Paneth cell or a stem cell. You cannot be both. However, if you gaze on a next gate here, you still see the green stem cells, you see the black, so the red Paneth cells, but you now see events where in the same droplet you have a green and a red signal. And if you now sort those, I hope you can see this, now you have 2 cells stuck together, and these doublets consist of one small stem cell in green and a large purple Paneth cell. Now I told you a while back that when we grow crypts in this cocktail, every crypt will grow forever. And we can expand them, we can turn them into billions of cells. If you take a single stem cell, it is very difficult. We need to try it 100 times to get one of the stem cells to grow. Toshi figured, well, maybe if the Paneth cell is an important helper for the stem cell, if I now take, rather than a single stem cell, I take a single stem cell with the Paneth cell together, one of these, maybe they grow better. That turns out to be the case. So now we're back to 100% efficiency. So in one well, we have one stem cell stuck onto a much bigger Paneth cell, as you can see here. And within a day, it's almost like embryonic development. They make a little cyst, the Paneth cell sits here. And then, as you can see, by day 7, we already have a fairly large structure. And by day 9, there's already several crypts surrounding the central lumen of the mini-gut. And what we see in movies is that every day one of the Paneth cells moves up, the next day there's a little gut, and then 2 days later there's a new crypt. So in that sense, the structures constantly make new crypts. And when there's about 30 to 40, they get too big, we have to break them up, they seal again, and it starts all over. Now, the referees are, I know that any of you has been ever involved in writing a paper and submitting to a journal, but referees always are extremely unhappy about your manuscript, and they have lots of complaints. In our case, it's always reviewer 3 who is particularly unhappy. And reviewer 3 said here that, well, these doublets, we were selectively sticking one stem and one Paneth cell together. So he wanted us to take a separate stem cell, separate Paneth cell, stick them together and have them grow. Now, technically, we could not do that. But Toshi could actually sort a few hundred of each, and in this case the Paneth cells are red and the stem cells are yellow. They come from different mice, and then when we keep them separate, nothing happens. But when we mix them, and you see here the mix, within 1 or 2 days this happens. It's rather striking. So particularly if you look here, the red Paneth cells, they're like shepherd dogs. They know where the stem cells are, the yellow stem cells, they round them up, and within 1 or 2 days they essentially rebuild the gut, which is rather, I hope it doesn't happen in your own gut anytime, but it actually is a great essay for us now to study how the stem cells and the niche cells, how they communicate with each other.